Okay, so welcome to a new session, a new talk in Introduction to Bioinformatics. And today with us we have Esther Lizano. Esther Lizano is co-founder and chief scientific officer of Flomics Biotech. This is a Catalan startup that has a business model around RNA analysis of blood samples. He also works as a researcher at the IBE, the Institute of Evolutionary Biology at ESCI-UPF. And Esther studied chemistry, like me. Actually, we were commenting about a long time ago. He did studied chemistry in the University of Barcelona, then did a PhD in the University of Leipzig about some enzymes called nucleotidyl transferase, tRNA nucleotidyl transferases. She did 2 postdocs, one in Max Planck in the Institute for Evolutionary Biology on the evolution of primate gene expression, and then another at the CRG back in Barcelona on brain transcriptomics in neurological disorders. And after these 2 postdocs, she worked at the CENAC, CRG, before joining the IBE and also before founding the company. So the talk today of Esther is called Perspective from a Basic Research Lab and a Biotech Startup. So thank you very much, Esther.
Yeah, thank you, Arnaud. That was a very comprehensive introduction. And have you seen, well, now that I realize I've been moving a lot during all my career, but now since the last 6 years, I'm working at the peer review building, which is in the Comparative Genomics Lab, which is on the left-hand side of the building. And then since the past 4 years, I'm also the co-founder and scientific officer in Flomics Biotech, which is very conveniently located right on the same level, but on the right-hand side, so I can just go from one to the other. But before starting into my topic today, I just want to make 2 disclaimers. The first one is that, oops, sorry, that's a bit dangerous. The first disclaimer is that I'm not here to sell you anything. So I'm going to talk about the research that is done in an academic environment and the research that we do in a more applied environment, so in a startup. Because you still have many years to go to start until you will start your professional careers, and there are like so many different options. I mean, you can go from like a patent office, you can start working in a patent office. You can be, I don't know, maybe in a hospital, in a genomics facility. You can join a big pharma or just a big biotech company. You can start, you can do like some science communication. We don't know. So that's why I'm going to talk about my experience, what we do in the lab, in the comparative genomics lab, in our startup. And the second disclaimer is that I'm not a bioinformatician. So it's not allowed to ask me which is my favorite programming language, because I don't program. Mostly, so I'm trying to make a sketch of like how big is what I do. And then I'm mostly in the lab, like developing new protocols, applying whatever is new to keep moving forward with our science. But as you can imagine, I've been working, I don't know, since basically next generation sequencing started, working with these technologies. So bioinformatics is super important. We are moving so much data that it's not possible to use Excel. So of course I need to use also bioinformatic tools. And what is also very important, what I realized during all these years, is that there must be a communication between the wet lab and the dry lab, because one without the other, they don't exist. There is no way that we can know if the method is working if I don't get feedback from the person that is analyzing the data, and they don't know what happened to the sample if they see like some artifacts. So this is like super important. So having said my 2 disclaimers, I'm going to first present you like the comparative genomics lab. This is a picture taken in spring 2022, and this is like a classic academic research. It's a big lab. There's like many people. We have PhD students, postdocs, technicians, some undergraduate students, and they also do some training that they do a stay in our lab to know to learn our techniques. And we even had like 2 American professors that were on a sabbatical. So this is like a very dynamic group. People come, people go, and we just keep moving. And also from all these people, like there is a mix of expertises. Some of them, they only just do like data analysis. Some of them, they are mostly focused on generating the data. And even some of them, they do both things. They go to the lab, they generate their own data, and then once they are done, they move to the computer and they analyze the data they generated. So it is not just a big lab in terms of people. We also are a big lab in terms of the numbers that we are moving. This is a summary that we did in 2021, and we were still like lagging because of the COVID, so we were still like going back to normal activities. So you see, we processed more than 1,300 biological samples. We did lots of extractions, libraries prepared. We do target capturing of some of these libraries. There is also a spin-off, which is the CryoZoo, that we are collecting samples from many different animal species that are in danger of extinction. And so out of these samples, they do cell lines, they do organoids, whatever is necessary to just help conservation in animal conservation. And we also, we participate in big sequencing projects. 2 of them, one is the ORG-1 project. This is sponsored by Nanopore. This project is about sequencing and assembling the genomes of any animal species that it's in the red list of critically endangered animals. And we also have a collaboration with Illumina and the CENAC, where we just send them samples, any primate sample that we can get, and we just sequence as much as possible. And I will tell you why. It's not just about sequencing because we can. So one thing that is common in academia is that we answer very abstract questions or conceptual questions. I mean, in the comparative genomics lab, the big question would be like, what makes us human? No, that would be like just quite an abstract, because one can answer this from many different fields. You can go to linguistics, to comparative psychology, primatologists, and what we do is just we sequence genomes. But we not only sequence genomes, we also try to understand the genomes, what is unique to humans. And to do this, we need to have data from humans, of course, but we also compare genomes to our closest species, like, for instance, chimpanzees, gorillas. And this way we can find what is unique for humans. So with all these samples, we do a lot of omics things. So like we assemble genomes, we have a PhD student that is specializing into assemblies. We do genomics, we do epigenomics, we do transcriptomics, and we are also even getting now into proteomics field. And then, as I was saying, we have to understand genomes, no? And you had Tyler here, I think, giving a talk about genome assembly. And there you could see now technology has been being so fast. You've seen always the classical plots of how like sequencing technology has been exploding, the prices are going down, all the algorithms that have been developed. And this is great. I mean, this is a huge resource, but not all genomes are easily assembled. It's not the same to assemble like a human genome with 3 gigabases in size than assembling a huge plant, or there are other animal species that they have genomes out of with 30 gigabases, and they are also fully repeated. So this is very difficult. And within easy genomes, there is also like more complications. Not all chromosomes are the same, easily assemblable, let's say. So this is what happens with the Y chromosomes. Y chromosomes, if you pay attention in genomic studies, they are underrepresented. They are always like left behind. And there are like many other reasons. There are 2 main reasons why this was happening. One is because of the evolutionary history of the Y chromosome. It just made it like nowadays it's like a big chunk of DNA completely repeated. So if you use technologies with, for short reads, you will just end up with like many, many repeated reads, which it will be, oh, sorry. So you will have like so many repeats and so similar that it will be like assembling a puzzle where all the pieces are of the same color and the same shape. So it's very difficult to do this. And another problem of the Y chromosome is the hemizygous nature, meaning that there is only one copy of the chromosome in the genome. So if you do brute force sequencing, imagine how much data you will need to generate to be able to have just enough data to assemble this gene. So there must be a way of just selectively capturing this chromosome instead of just throwing a genomic library into a sequencer. And also like getting like longer reads. And then to leverage these problems, we took, there was a technology that is already working since many years in the lab that is like fluorescence activated cell sorting. And this, what you do is you stain your sample depending on the characteristics. And you can see here in this plot, that's where the Y chromosome relies. And this goes through a series of fluidics technology, and a laser is just separating your Y chromosome from the rest. So we ended up with having like a tube with only selectively the Y chromosome separated from the rest of the chromosomes. And that was already done in another group. They applied this technology, but they sequenced with Illumina and PacBio. So what we wanted to do, it was like sequencing with nanopore to have like as long as possible reads to be able to assemble this genome more easily instead of having very small and repetitive pieces. So after doing the chromosome sorting, we had to do, the problem was that since these chromosomes are stained with fluorophores, this interferes with the measurements to know the quantity or the quality. So we had to devise a method to purify them and to be able also to concentrate it, because out of the cell sorter, like it comes like basically in liters. So we had to find a way of just concentrating this, and then after concentrating, we were able to do 4 min-ion runs. By now you know the technology, nanopore. And we also took a little bit of the DNA that we extracted to do some Illumina sequencing to be able to polish the assemblies. You know that there are like many, like nanopore still has like some errors in the sequencing. So what we accomplished with that, it's just like, as you can see, this is the coverage that we obtained for all the chromosomes, and we had like a huge increase in coverage of the Y chromosome compared to the other chromosomes. And you might be asking why we have other chromosomes if we were selectively. And this is like something very important. It's like there is no perfect data set in biological sciences. There's always some artifact or something that you have to live with it. And the problem with this is like, especially we were getting quite some chromosome 22, and it's because chromosome 22 is also very close to the Y chromosome, and when you separate it, you also separate a bit of the chromosome 22, or because they break into pieces, it's impossible to really be able to get like 100% enrichment of one chromosome. But this is already like a huge improvement compared to doing like brute force sequencing. And here we have like a depiction of this one here. This is the chromosome we assembled compared to the Y chromosome from the Human Genome Project compared to the gorilla assembly. And so what we managed was to get like a contiguity which is like 2 orders of magnitude bigger than or higher than the gorilla assembly. And also, as you can imagine, this was published in 2019, and all these projects, they move slow, no? Because we had to find a way of cleaning them up. We found some problems, then we got the data, we sequenced, we wrote a paper, We went into peer review, it went back, and and at some point it was published, the paper. So what happened during all this time, while we were waiting for the paper to be published and so on, we had already a tool for sequencing Y chromosomes, and a new project just started, which was like sequencing many different Y chromosomes from human populations to just reconstruct a topology of the Y chromosomes across all these different populations. But while, as I was saying, this takes very long time to process everything, while we were working on one side, it just happened, so we were starting with the chromosome sorting, like doing our own method. But I don't know if you ever heard about the nanopore and adaptive sampling technology, no? So this is something recent that a nanopore is developing. Is still not perfect, but at some point it will be like very helpful. What you do is you put your genomic library without any enrichment, without anything into the machine. And then you tell the software, okay, whenever I'm only interested in this part of the genome. I only want from this coordinate to this other coordinate. So when a molecule is entering the porus, it starts sequencing, and then the software decides, is it what I want or not? If it's a molecule that we are not interested in, it just takes it out of the pore and then starts again until it finds like one molecule that is what we want. So, and then this keeps sequencing and is separating. Like you get a bin with the data that is not interested and the data that what we want. So one project that started the intention was to study Y chromosomes across human populations. Now it's also turning in the same. Plus, we are doing a comparative analysis of our own chromosome sorting methodology compared to this adaptive technology from nanopore. And by the way, I have, I don't know, because probably you heard many times about nanopore. I mean, I brought you, maybe, I don't know if you ever saw it before, but then we can, afterwards, whoever is interested, I can show it this little thing. And that you can see that it is really true that we can take them, these machines, almost anywhere. Yeah, so that was a way of getting better data to be able to compare ourselves. But as I said before, there's also, to know what is unique to humans, first we need to know what is unique to other species, for instance, to chimpanzees, our closest relatives. And for this we need data. No? Of course, again, we need to know what is different among chimpanzees and then compare it to humans. So we got into a collaboration with the Max Planck Institute in Leipzig, where the primatology department of that institute, they had a program which is called the Pan-African program. And what they do is they study wild chimpanzee populations in terms of culture, behavior, ecology, and genetics. And that's the part that we enter, the genetics part. So what these people did, like along many, many years in going to trips to Africa, they just collected a huge, they have a huge collection of fecal samples. Fecal samples, whoops, from across all the chimpanzee range in Africa, all 4 subspecies. And what they told us is like, hey, we have all these samples, do you want to analyze them? And I said, yes, of course, we really want to go for it. But one thing is like, it's fecal DNA. Because, I mean, this is fecal DNA, it's a great resource to study wild animals. Because for obvious reasons, I mean, it's not so easy to go to Africa and just ask a chimpanzee, give me a bit of your blood. This is an obvious reason, it's not so easy. But the second is, and most importantly, is because of ethical reasons. We cannot just go and sleep the animals to get a sample. So fecal samples are actually a really, really great source of DNA. And not only this, we also know, like, each of the samples, they come with coordinates. So they tell us this was taken with this latitude, this longitude. We know exactly where it was picked up in Africa, the sample. So I said non-invasive samples are a great resource for DNA, but they also come with their own limitations. The main problem with fecal DNA is that the endogenous content of DNA of the animal that it came from is very low. So most of it is microbiota from the own animal, or also because when it's deposited on the floor, like also other fungi and other bacteria are growing there. So we don't know how long it's been exposed exposed outside in the wall until somebody came and picked it up. So what we did first, it was to measure how much chimpanzee DNA is in each of the samples. And you can see here that most of them, they have very little endogenous DNA. They are below 1%. So the amount of DNA that is in the sample, it's critical to have a successful sequencing. So it's again the same. If we do like a brute force sequencing for a sample that is below 1%, imagine how much we would need to sequence to have like a 1x coverage of the chimpanzee genome. This is like economically not viable. Not viable, and also like we would have like so many teras of data that would go to waste that we don't have even space to store it. So what we thought is like we have to find a way of getting still this data out of the fecal samples. So we had these more than 800 samples coming from 48 different sites in Africa. And what we thought is like, okay, we are not going to do brute force sequencing for the whole genome. We are going to selectively capture or enrich for two different parts of the genome. One was the chromosome 21, and the reason is because the chromosome 21 is very small, but it's informative enough to do genetic studies. And the other part that we were also capturing was the exome, which is like all the protein coding genes in the genome, which is also a very small part. It's like about 2% of the whole genome. But that came with another problem. We said, okay, perfect, we are going to do this capturing, so then we are only sequencing what we are interested in. But target capturing is very tedious, it's very slow. And it was like no way in the world that the PhD student that was working on that would have time to capture 800 times the chromosome 21 in one hand and the exome in another hand. So we thought, okay, yeah, we just pull the samples. We just pull the samples, and if instead of doing like 1,600 captures, we are going to do 300, which is like in terms of time, like much more available. But to do this library pooling, it means we had to modify our own library prep methods to be able to separate one sample from the other, because if you put all together in one tube and then you don't tag them uniquely, once you get the data, you don't know from where it was coming, this data. So we just went into the literature for ancient DNA, because that's not a problem that it was new. So we just adapted like their methods to our own problems. But of course, before moving into like the big project, we said, okay, we've never done this before. We also designed our own base for the capture for the chromosome 21. So it's like, let's make sure that this works. So before we knew, we were already like, we got into a new project, which was like a very technical project, where what we did was like, we designed an experiment taking only a subset of the samples, so only 60 samples out of the 800 and only from 15 sites. And we did different mixtures or different experiments to tackle quite a few technical problems that we were trying to solve. And among them, it was like, what about the sample quality? Because fecal DNA, as you can imagine, it has like way worse quality than a DNA sample that comes from a cell line. We also evaluated the starting DNA amount. Maybe by putting the double that is normal, we will be able to capture, to retrieve more data. Also the hybridization number, like how many times can we just repeat and repeat experiment to get more data out of one sample. And also very important, replicability. Because also like if you cannot replicate your own experiments, it's completely useless. So I'm not going into the details for what we went and all the all the parameters that we evaluated. But what this big project first it turned into a smaller one, which we published. And in this paper, what we published, it was like we established a set of guidelines for non-invasive targeting of resequencing studies. So once we had our guidelines, now we knew how to do all the experimental work. This Claudia, the PhD student, she said, okay, now I can go back to the lab to my 800 samples and now start producing data. So that's what she did for quite a while. She was just capturing and capturing, then everything was sequenced. And then she went back to the computer and started analyzing, and you can imagine with 800 samples, you can do lots of analysis. That's what she spent a lot of time analyzing, and among one of the main results of this study was like she was able to reconstruct the chimpanzee genetic history from the middle Pleistocene, so 2 million years ago, up to today. And then what she reconstructed was like from the split between the bonobo and the chimpanzees, like which migrations they had, which bottlenecks they suffered during all these 2 million years until nowadays, and how all these migrations and bottlenecks, they affected how the diversity of the modern day chimpanzees are. So as you see, it's like quite conceptual. We are like interested in knowing how chimpanzee genetics is. It is like nothing like super applied to today. I cannot sell this and get money out of it. But there is this very very academic part, but there is also like some spin-off in the paper, which I think it's an application which is like very interesting. There is a huge problem with primates, which is like the illegal trafficking and poaching. This happened in 2017, like where 2 baby chimpanzees were confiscated in Nepal. These 2 animals, these baby animals, they were captured and smuggled up to Nepal to enter into the black market. They can get very expensive in the black market. So a problem is like if you see these baby chimpanzees, like you don't know where it came from. You don't know if it's a central chimpanzee, Nigeria, Cameroon, or what species. So you cannot find the country that was allowing these things to happen. So by sequencing all these many samples, that as I said before, that they were geolocalized, we constructed a huge data set of, sorry, not a data set, a database. So now, and there was another postdoc that he developed a method, and then what we are able is like to geolocalize the origin of a given animal. You give us like a sample, it doesn't matter if it's a fecal sample, a hair, some blood, we will sequence it, we will look into the database, and then we can tell where the animal comes from. And we do it with an error of 100 kilometers. With 100 kilometers even, so what you have here, this is for very low coverage, so only chromosome 21, so above 0.25%, which is very little of the chromosome 21, we already have the error of 100. We are already like within 100 kilometers. And this part here is also like if we have contamination, because these samples are manipulated by many people. People go to the field, they put it into the tubes, and then it can be contaminated. So it's very difficult to separate a chimpanzee read from a human read. So even if we have very high human contamination, we are also able to place where the sample is coming from. And with this, we hope that governments, they will reinforce some laws and some whatever it is necessary to avoid that this is still happening. And then, of course, I mean, as an academia, very important, publish papers. So this started in 2016. 2022, we got the paper. We made it, Claudia made it to the I will say, portada, to the cover of the magazine. And as you can imagine, it's like a huge study with many different results and many implications. And if you are interested in seeing this, but you don't want to read the whole supplementary, which is like maybe 80 pages long, just the supplementary, you can go to Claudia's Twitter, and she has a very nice thread pinned where she's like summarizing the main results of this work. So now moving to different or similar topic, but different kind of samples. As I said, no, we compare our genomes to other genomes, and what is a huge repository of biodiversity are natural history collections, so the museums. Museums are full of samples. And then we said, okay, let's start collaborating with museums. If they would be willing to give us some samples, we can sequence them and also include them in our studies. And the work I was presenting before with chimpanzees, we also do it with gorillas, with orangutans. So we are really expanding this work to the whole primate clade. So, yes, I was saying, history collections are a huge resource for us. We build collaborations with them. We go to the museums. We are sampling here. Here, these are 2 PhD students. I went to Brussels, and then they were taking samples. And then we have, at the PRBB, we have a clean room. We have a clean room to process these samples. And this is very important that we do it in a clean room, because I was saying contamination is a huge problem. It's very difficult. If you have a sample contaminated, you won't be able to separate these reads from the human reads. So it will be wasted. So we have the natural museums. We have our own labs. So we got into it. It's like, okay, let's get more data. And what we are trying to do is not just getting samples and samples because it's possible. What we try to do is, like, since we have like the modern day samples, these populations, they have decreased during the years. There are like some areas in Africa where maybe 100 years ago there were populations of chimpanzees that are now, they don't exist anymore. So what we do is, like, we try to complement the modern day samples with museum samples. So it's like, do you have samples from these regions? And then we take these samples whenever is possible. So it will be like also a way to study how these bottlenecks, they were also, how was the diversity 100 to 100 years ago of the chimpanzees and how it is nowadays. But as I was saying, there is no perfect data set, and it's very important before getting into the real project and sequencing, spending a lot of money. It's like, first you need to make sure that what you are doing is correct, is right, no? That your data is, it will be usable. So before starting with the hundreds of samples, we did a small pilot project, because that was also something new for us, where we just took, I think it was like 35 samples, and we did very low coverage shotgun sequencing. And we look at different quality parameters. One is like, I don't know if you know that, but ancient DNA from DNA from the moment of death, it just starts degrading. It gets shorter and shorter. And the older it is, the smaller pieces we have of DNA. So one parameter is the library length. After we transformed the sample into a sequencing library, we didn't fragment this DNA, and then we saw that the library, you see the peak is around 200 nucleotides, so that was already like a good thing. It's like we are having small libraries. Then we sequence these libraries, and we check the DNA fragments. We remove the adapters, and then we have also like the peak is around 80, 90 base pairs. So that was a good indication that when we were sequencing, it was like really the DNA that was contained in the sample, that we were not sequencing our own contamination, because this could happen as well. We also look at the other quality parameters like duplication number. It's very important not to have many duplicates, because if your library is very duplicated, you will just sequence a lot of data that is unusable or uninformative. If you have 1,000 times the same molecule, it's not. So, and then we also estimate how much DNA is in these samples. Some samples have very little, like 0.3% of endogenous, and some samples have a lot, 51%. And this shotgun, this low coverage shotgun, it also serves us to separate which are like the good samples, are the ones that we can, they are worth, that we can get even more information, and the ones that we can just sequence over and over again, from the ones that are not worth sequencing more, because it doesn't matter how much you sequence this library, you will not get new information. And then another parameter that we wanted to look into these samples was like in ancient DNA or historical DNA. This is not ancient because they are not as old, so we call them historical DNA. It's like old molecules, they accumulate some damage, and this accumulation is mostly at the 5' of the molecule and at 3' of the molecule. And the modification is, it's called the amination pattern. So what happens after death is like the cytosines, they lose an amino group and they convert into a uracil, which in turn, this uracil, when you sequence it, it will be read as a T. So that's what you are seeing here. Like, this is like from the 5', the distance of the first position, to the last position, there is an accumulation of T's, and this is like the complementary, it's an accumulation of A's. But here I'm cheating a bit because I took the best sample I have. And first, when I look at these deamination patterns, I was very disappointed because they were flat lines. They were flat lines because I was using software that is thought for ancient DNA for very old samples. So then I thought a bit, and it's like, wait a moment, my samples are not that old. So what I just had to do, it was like to zoom in. So I just, instead of having like a huge Y axis, it was like, let's look only in this. And then, there it is. There was a little bit. But of course, this is not a good quality parameter that we could use for these samples. And also because we have like, that is the one that had most damage. But then it's like you look in the literature, and then like someone else had this problem, and what this researcher, Pontus Skoglund, he developed an algorithm to measure this postmortem damage, and he made a score to evaluate the damage of a sample. And the only thing that he's doing is that he's taking all the sequences, and it tells you the probability that this sample, this sequence is old or it is not old. And then depending, so if you are below or more, or 0 to above, it means the likelihood that this molecule is modern is very high. And then the older and older the sample gets, this you move into this part of into the graph. So when after we ran this program with our samples, we saw that they actually fit into like the same category, age category as our samples. And then, so that's, after doing all these quality controls, that's when we said, okay, we are good to go now. We can keep sequencing. And then the final quality measurement before going into the big sequencing, the final one was that we wanted to see if we were able, with the data retrieved from museum samples, we were still able to separate the 4 subspecies of chimpanzees. That's what you have here. So each color is one of the 4 subspecies. The dots are, what we did is in this principal component analysis was to compare our data from museums with modern day samples from blood. So the dots are blood samples, and the crosses are the museum. And it was amazing to see that actually with very low coverage, we were able to separate these four subspecies. But if you pay attention here, these two, these are the central chimpanzees. They are not really clustering together. So we don't know yet. This could be biology. Maybe something happened in central chimpanzees, or could be like a complete artifact from our data. So you have to be very, very sure before claiming anything that what you are saying. So it's not that we found like a new subspecies, no. Just first you need to understand what is going on. So what we are doing now is just like we are in process of sampling. We have 12 more museums involved into this. And then we are generating more data. And then finally, so just we are moving into even older samples, which is like paleoproteomics. Paleoproteomics is just like sequencing proteins from very old samples. We know that, so DNA, the oldest one is like around 1 million year That's the world record for DNA. But if you can see, for proteins, the oldest, the world record is at almost 4 million years old proteins. And not only this, this sample was from Africa, so from very temperate climate, which it opens a lot of new possibilities. And what we are now doing is like trying to see, the big question is like how further we can go back in time. I'm just going to go very quickly because this is like a very long process. But we go from like the original sample that a paleontologist is unearthing. They claim, they think it is one species or another. We do proteomics work, just basic double tandem mass spectroscopy. From this, we are able to retrieve and reconstruct protein sequences, and out of this, we can just do whatever analysis you want to do. And in the case of Gigantopithecus, actually what it was possible to do, it was place it into the, to place it phylogenetically. And then it was observed what the paleontologists thought. It was like, it's an extinct cousin of the orangutans. So as you can see, now I'm done with basic research. What you can see is like we just do conceptual questions, very abstract or not really applied. So what we do in Flomics or in a startup, it's completely opposite. We are really like, we have to find an application to the work we are doing. And the first thing, also like the very first difference is that we have to be corporate. There is already a template for the presentation, so it's like you have to follow this template. And deep within, the technology is the same. There is no such a big difference from academia. We take a blood sample, we do an RNA extraction, we apply some machine learning algorithms, and then we find a signature for cancer. And we do it in a way that we can separate controls from patients. So until here, it wouldn't be a big difference with academia. The difference is that in academia, you would just say, hey, I can separate controls from patients, I publish a paper, I'm done. Then I move to the next thing. In a company, it doesn't work because this doesn't give you money. I mean, publishing papers is like you pay for that. So what it changes is like you need to have a problem, which for Flomics is like cancer is detected too late. And because of this late detection of cancer, it poses a lot of problems to the system and to the patients. It's a lot of money, the chances for recovery are very low. So if we can find a way to detect people that are beginning to develop the disease, it will be so much better for the patient and for the system. So we have a problem. You have to provide a solution. So which is what I was saying. You have an individual that is at risk because of age, because it's a smoker, because of the family history. Then we do a non-invasive screening just from a blood sample. We can test for 5 different or the most common cancers, which with the results, you can target diagnostic tools. You can say, okay, maybe there is something in your bowels, do a colonoscopy, or you are fine. You can just go and next year you do again your own, you repeat this analysis, like a blood analysis, which it will be like in an improvement of quality of life. So we have a problem, a solution, a technology, but it is not only this, this solution, it has to be scalable and turn into a business model. It doesn't matter how great your idea is, how good your algorithm is, if like nobody is interested out there, you will not sell it and you will not make money, your investors will be very angry with you. And also, what it needs to be done is like not only 5 people are interested in my technology, it's like as many people as possible, because what I want to do is like grow this business and be successful with the company. So, of course, as I was showing at the very beginning, the people in the comparative genomics lab, of course, in Flomics is not only one person there. We have a great team of people, and not only we have time for work, we also have fun. It's also very important. It's like one thing is work, one thing is your private life. This is like you need to separate it and have your own private time. And just to finish, I want just like maybe for your questions afterwards, it's like this is only a comparison of similarities and differences between both worlds. So this I repeated many times. In academia, it's very abstract. We answer questions, and by answering these questions, we get new questions that maybe somebody will take this technology or these things that we are studying, and then they will find an application. That's what we do in Flomics. Also, in academia, things are slow. Since you start a project until you publish it, it can be like many years. In a company, it's fast. You need to change. As soon as something is working, you apply it. If it doesn't work, you forget about it. Also, the knowledge in academia is very important. Your methods, your algorithms, like to make everything peer-reviewed and so on. In a startup, the knowledge is the product impact. How is this going to impact the product I'm trying to sell? And then, of course, there are similarities in both environments, like collaboration. You do nothing if you don't collaborate with other people. You need the knowledge of your coworkers, of other institutions. Both places, they promote creativity and innovation. It's like all the ideas are welcome to test or to try if they make sense. And then it's a great place for continuous learning. People who like to learn continuously, that's like the best place you can go. So, yeah. I said I didn't want to convince you of anything, of doing academia or anything, but we are always looking forward for brilliant people, motivated people that they want to join us. So if while you're studying there is a field that you like, it doesn't need to be like in these labs, like any other lab, just ask your students, ask your professors to open doors that if you want to do an internship, you want to do your master's degree or whatever you want to do. Yeah, thank you.
Thank you very much, Esther, for this great presentation. So now time for questions. Yeah.
Well, it's there, or do you think it will be a machine that will let you transport the data gens without losing the information?
Sorry, to transport?
Like, to transport the data gens, like the gens that, well, yeah, no?
What do you mean,
like
the genes or?
The genes, yeah. Like, without losing like the information of the genes, like as you said, like at the beginning, like when you transport like the genes of the monkey, no, the orangutan, I think that you should tell, like you lose some information.
Ooh. So far, I mean, there is like no perfect technology, no perfect data set. So when new next generation technologies, they started, we thought that was the answer to everything in life, because until then it was done by hybridization arrays, and then we thought, wow, this is great, we are just sequencing. It is moving very fast. It's crazy. Like 5 years ago, I wouldn't think that one can just sequence native DNA like in a nanopore, that I can sequence DNA, RNA without doing any PCR or anything. Good question. I mean, that would be amazing. But yeah, that's for all the new generations to work on this and try to get this technology.
I wanted to ask, do you think is it worth it to use non-invasive samples in all biological experiments despite the inconvenience?
To stop doing biopsies and all this, you mean?
Yeah, I mean, in the most experiments or in
Of course, I mean, yeah, that's what we are trying to do. We started like many years ago in our lab. Most of the samples, they were from blood or tissues, but this had to be from animals that died, and then they get a necropsy, and then we get a sample, or they are put down for a medical checkup, and then we got the blood. So that's why we are moving into non-invasive, and we are not. I talk about fecal DNA, but there is like from hairs, from blood, which is semi-invasive. Urine, there are also some papers, some people who were able to sequence out of urine. Any biological fluid that would be, it is possible. Yeah, but what we have to get better is like in separating the DNA from that we are interested from the rest of the DNA, because it is in a very slow and very small amounts. Yeah.
Do you think that the work you have done with chimpanzee can be done with more species, or is more difficult?
No, we are doing it. We are doing it from chimpanzees, gorillas, orangutans, ferrets, the Montseny brook as well. Technically, it doesn't matter which animal it comes from, it's DNA. So we just we can apply this to any other species if you are interested.
Thank you.
Yeah.
Hi. So you talked about using fecal samples from chimpanzees to extract DNA for ethical reasons, and my question is if there are other methods that do not interfere with ethics, and if they are, do you think that these other methods are as effective or more as the one you used?
In general, of course, the most effective methods is this. It's like getting a carcass from an animal that was just died because of natural reasons, and that also happens. Like researchers are in Africa, and then they collect that and they freeze it, and then they send it. The animals from zoos, but we only get the samples when they need some medical treatment. So then we take advantage of this. That's. I'm not sure I answer you.
Well, no, yes.
Yes. Oops.
How long do you think that huge projects like the Illumina project will take, and do you think that it will finish at some point, or will always be in progress due to the exponential data growth?
Well, the Illumina, it's huge. It started, I think, in 2017, 18. I mean, I'm not mega involved in this project, but we made the phase 1 when at some point we said, okay, stop it, and we already analyzed this. This is about to be published soon, and not only our paper, but like many other accompanying papers. Imagine with having like hundreds of genomes of also hundreds of different species, it's like you can do a lot. Then we started the phase 2 that was stopped this summer, and now we are trying to see if there will be like phase 3 or not. This we don't know. But of course, I mean, as long as there are samples, we could just go on and increasing the data.
There's any way to analyze the data from very ancient uncontaminated samples, like the history samples, in the DNA is highly degraded, or there's a way to increase the quality of the DNA?
Yeah. So DNA, there is a limit, there is a limitation, and there was a paper that they said where they calculated what was like the maximum age it would be possible, and they calculated it was around 1 million years old, because as I said, like DNA goes like degrading and getting smaller and smaller, and there is a point that is like so small fragments that we can sequence them, but then it's impossible to know where it's coming from, to map them. So DNA, as far as we know now, it is like already at the maximum. That's why we moved to proteins, which is proven that we can go further in time.
Yes. Can you tell us more about what is unique for chimpanzees in comparison to humans?
Yeah, that's a great question. We still don't know what makes us human, of course. What we have, thanks to all these projects, we have a catalog of variation. Now we are increasing our knowledge in what is, at least at the DNA level, what variations or what duplications or deletions we find only in humans compared to other. And then by the Illumina project, by sequencing, because we thought like some parts were unique to humans, but by sequencing many other primate species, we've seen that chimpanzees do not have it, but the other have it. So actually, humans, we are not so unique. It's like we decrease what we thought instead of making it bigger.
Hi. When you were talking about DNA samples, you said that 1% is so little DNA. I would like to know how many DNA is needed, like a minimum percentage.
It is needed very little. I mean, all the lab methods are very powerful, so it's so little needed that you cannot even measure with the technologies we have now. So if you do a nanodrop or a qubit, it will say you are below the limit of detection. But as the Neanderthal sequencing project they proved, I mean, you can just believe that there is, because you cannot measure it. I mean, it is there. The problem is that you cannot really measure. So you just do a PCR. That's why, I mean, PCR is like our biggest resource, to be able to amplify whatever is there and then to sequence it. So as long as you have one molecule, theoretically you can sequence it.
Hi, I just want to ask, do you think in humans we can use fecal samples to detect cancer or some diseases, like you did with chimpanzees and others?
Sorry, I'm not following.
Do you think that you can use fecal samples to detect like cancer or disease in human?
I think so. It's just a matter of, definitively, I mean, maybe not all cancers, but some of them, like colon cancer, what you are sequencing are the epithelial cells of your bowels that are like coming out, with the sample. So if there is like a signature that is there and you can pick it up, I think it would be possible. Another thing is doing it like through RNA, like we do in Flomics, because RNA, you know that it's way less unstable and degrades more easily than DNA. So, but at the DNA level, if it's possible, one can get into it and do it. Or microbiome, for instance. Like one could look, instead of looking for your DNA, look at how the cancer is modifying your microbiome and try to figure out a difference between that.
How far we can go back in time to analyze samples?
So DNA, 1 million, but it has to be in like very precise conditions. So this sample that was aged or dated at 1 million, that comes from Yukon in Canada, found in the permafrost, so like frozen for many, many years. So the DNA was like very well preserved. With proteins, we don't know. We don't know because I was saying like proteins, the good thing is that since this sample with almost 4 million years old that came from Africa, that was exposed to the sun and to other extreme conditions, and they were able to sequence peptides. So that's what we are also doing now. We have samples in our lab up to 12 million years old. So the question is like, how much can we push the methods? And not only the lab methods, but also the analysis. Because in protein sequence, to retrieve the sequence, you don't do it by reading it. You do it by doing a database, by doing matching with a database. And how do you do a database of protein sequences that you don't know how they were 12 million years ago? So we are taking the Illumina project from many primates. We are trying to figure out which parts of the proteins are the ones that give us more information to be able to separate the samples.
It's about like when you start, you make a lyric question like, what makes us human?
For instance.
And my question is, do you have a possible answer to this question?
No.
No.
No. Not at all. I mean, once I heard a great geneticist saying that something that was unique to humans was like this curiosity, and then also not just the curiosity that, or we are a bit crazy, like somebody got into a boat many years ago with like a very basic boat and just crossed an ocean. Some other people got into a rocket and went to the moon. So this could be one thing, but what if we have a gene for that? This I don't know. It's like, it's not just one simple answer. I think it's like a huge mixture of many, many things, and throughout all the evolution that made us human compared, because at the DNA level, we are almost the same as any other primate.
Okay, so let's thank again Esther.
Thank you.
And if you want to see the nanopore, just come here closer.